Professor Yamamoto has had several days with us recently, inspecting our work. So I hope we meet with to your satisfaction. I was asked to speak to describe the Singapore National Precision Medicine Program because we think it is potentially a valuable collaborative scientific partner in state-of-the-art international cross-population molecular and population epidemiology programs. I'm going to start by setting up the motivation for the work. One problem that Singapore faces, like many other high-income countries, is it faces an epidemiological transition, the demographic shift of an aging population. And as a result of that alone, the national projections are that there are going to be substantial increments in the burden of major chronic diseases such as diabetes, heart attack, cancer, and stroke. Currently, healthcare accounts for a relatively small fraction of healthcare spend, but the healthcare budget is expected to roughly double over the next 15 years if healthcare costs aren't constrained. And so there's a tremendous interest in trying to understand what are the determinants of disease for the Singapore population, and how can we use state-of-the-art diagnostic, prognostic, and therapeutic strategies to bend the curve and improve health outcomes. Now, the middle panel here shows a very interesting phenomenon, which is that not all populations are the same. Different populations have different exposures and they have different health outcomes. And the middle plot shows the different incidence of diabetes among South Asian people compared to European people, with a 3- to 4-fold higher risk of diabetes amongst South Asians compared to Europeans. And this is the bread and butter, this is the lifeblood of epidemiology. Different population groups with either different exposures or different outcomes as a fertile ground for discovery. And here Singapore is very well positioned because we have 3 major ethnic groups, the East Asians, Southeast Asians, and South Asians living side by side, but with still living historic, traditional ways of life. So different environmental exposures, different behavioral patterns, and different genetic backgrounds. And so this becomes a paradise for exploring exposure outcome type relationships. The third motivating factor behind setting up the program of work is shown on the right-hand side. It's a well-known figure. It shows the lack of genomic data in the world's global genomic data sets, with Asian data representing still just a minority. And if there's missing genomic data, that also means there's missing metabolomic, proteomic, transcriptomic, and other population phenotypic data. So it's not just the genomic data that's missing, it's the whole population cohort data that's missing at scale. So to advance the healthcare need and to advance the science, Singapore has strategically set up a 10-year research platform, which we call the National Precision Medicine Program. Set up in 2017, 10 years, 3 stages. Each step being, the first step being a small one, the next step being an important moderate one, and the third step being the biggest and last. In the first step, it was really pilot work. It was to say, well, okay, can we put together a cohort of 10,000 reasonably well-characterized individuals? Can we do whole genome sequencing? Can we do proof of principle linkage to the EHR to demonstrate that people can work together, generate high-quality data, and we can recapitulate the things we'd expect to see in 10,000 individuals, the expected associations. That was phase 1, 10,000 people. It's now done, and I may show some of the results of that work later. We're now in phase 2. Phase 2 is what we call a proof of value scale, where we're going to put together a cohort of 100,000 people and use that to demonstrate discovery, show that if you have a cohort of 100,000 multi-ethnic Singaporeans, that can drive innovation and discovery and healthcare change as proof of value. And if that works, then phase 3, which we're starting to plan for next year to start in 2 years' time, phase 3 will scale up to approximately about 1 million Singaporeans. That is, if you like, given the size of Singapore, only 3 to 5 million people, that represents basically a whole population scale-up model, in which case, we're aiming to bring precision medicine strategies and precision medicine research to population scale. So we're in phase 2, and what do we plan to do in phase 2? It's got three main components. It's got building a cohort, it's got doing molecular sequencing, and it's got linkage to the electronic healthcare record. I'm going to talk to those three in turn. The cohort, SG100K we call it. It's a prospective, longitudinal prospective study of Singapore's multi-ethnic Asian population. It's a general population sample, 100,000 people aged 30 to 84 years, male or female, with three major ethnic groups, Chinese, Malay, and Indian, with some oversampling of Malay and Indians to improve study power. We've designed the study to be internationally, not just competitive, but perhaps more importantly, internationally aligned. We've looked at best practice in the world, in global biobanks. All of us study the UK biobank study, the German study, the Finland study. We've also looked at Taiwan Biobank and Japan Biobank, and we've tried to choose tools that are broad, so get us across the whole physiological and biochemical spectrum, but at the same time are also interoperable, so that we know if we measure something using this particular approach, we'll be able to then make international comparisons accordingly. There are four domains that participants are characterized in: set of questionnaires, which are health and lifestyle, cognitive testing based upon the UK Biobank cognitive test, food frequency questionnaire, validated for local food intakes. Physical measurements, fairly standard: height, weight, 12-lead ECG, blood pressure, spirometry, arterial stiffness by Vicorder, accelerometry, the activity device as per the UK Biobank, worn on the non-dominant wrist, and a functional treadmill test. Quite cool. We're doing some imaging on all participants. We've got carotid atherosclerosis being quantified, subclinical, preclinical atherosclerosis being quantified by IMT using 2- and 3-dimensional carotid ultrasound. 3D bit gives you plaque volume. We've also got extensive retinal imaging on the basis that this is a great place for you to see the nervous and the vascular system side by side, and of course the microvascular system using a technique called optical coherence tomographic angiography. So you get to see the microvascular system, which is critical, of course, in many chronic diseases, particularly cardiovascular, metabolic, chronic kidney, and cognitive disease. Right-hand side, DEXA to quantify bone mineral density, body fat composition, body muscle composition. It's important assessments of sarcopenia and adiposity. Biological samples, blood, serum, plasma, and RNA as per biobank standard. We additionally collect stool for microbiome on approximately 40% of volunteering participants, and skin tapes, so we can assess skin metabolome and microbiome for all participants. We've got a fantastic cloud-based data infrastructure that allows us to collect source data directly from machines so that we're confident it's high quality. And the consent is fit for purpose in the sense that it allows all the 21st century kind of things that we'd expect: sample and data sharing, molecular phenotyping, linkage to EHR, recontacting people by genotype and by phenotype, and working with industry both local and internationally. And just as an aside, we've tested the sort of the recontact by genotype and phenotype mechanisms, and we get very good engagement from the population. We get 50 to 70% of people happy to come back for further sub-studies, which is fantastic. So I talked about how does this cohort fit within the precision medicine program? Well, it represents the cohort that's going to be used for the phase 2 precision medicine program. And so on to the next two parts, the molecular phenotyping. The key molecular phenotyping being done at this stage is whole genome sequencing. This is being done as a public-private partnership with Illumina that was announced in May of this year. The ambition is to complete 100,000 whole genomes by 2025, so 2.5 years from now. We started the sequencing in June. We've gone through the first 5,000 people, and at scale we'll be doing 30,000 genomes per year. It's being done using the NovaSeq platform with DragonX calling to generate 30x genomes. We're also planning to do DNA methylation profiling by methyl epic or equivalent. Probably won't be methyl epic, it's a bit old, but some of the more modern capture methods look very attractive. As I say, we've sequenced 5,000 odd samples now. It's almost getting to the point of being real-time DNA sequencing. It takes, on average, from the time we delivered whole blood to the extraction facility, It takes 60 hours from sample delivery to sequence data being generated, giving the possibility for some interesting downstream experiments based upon real-time feedback of genotypic information. The third chunk of data that we're bringing into the precision medicine effort is linkage to the Singapore National Electronic Healthcare Record and other governmental records relevant to health. So in terms of the electronic healthcare record, there is a single central repository for electronic healthcare record that's managed by the Ministry of Health. There are approximately 140 institutions that feed in their data to that platform, and that predominantly comprises the public healthcare institutions, the government-funded state system that is responsible for about 80% of clinical care in Singapore. In addition, a small number of private hospitals that account for the other 20% contribute their data, but we're working to extend coverage of the EHR to include private hospitals in due course as well. Clearly, with 140 different data providers, the data come in somewhat in different formats and with different degrees of QC and cleanliness. So there is a core team at MOH that's responsible for some QC and some mapping, so that and the coding, the overall coding is done using the OMOP scheme, so that the researchers won't have to wade through 140 different EHR data sets. They'll be provided with a coded, mapped, OMOP mapped data set to enable harmonized researches against the EHR. Singapore are completely committed to making the data available not just to local healthcare researchers and clinical researchers, but also more broadly. We've already started to make our pilot data available in a democratic kind of way, as aggregate data through a series of genomic web services. One called the Chorus browser that allows you to look at allele frequencies and annotations in a population-specific way, aggregate data, but broken down by Chinese, Malay, and East South Asian. Platforms that allow you to do imputation on your array-based data, or to look at polygenic risk scores if you input the genotypes. They're not open, but they're perfectly. You simply have to sign up for an account, which takes a day or two, and after that you can browse, you can put in targeted queries. You can extract small chunks of data democratization. It's a first step. We're also committed to making individual level data available. So I've talked about the research phenotypes and the molecular phenotypes. I talked about the clinical data. Those are going to be merged within a bespoke platform being set up by the Ministry of Health called the Trust Research Data Analytics Platform, hosted by the Singapore government as a single source of truth to go to for access to research data. That data lake will be made available on application to qualified researchers to pursue relevant biomedical research. The expectation will probably be that it'll be widely available within Singapore. The data contributors to the platform, such as ourselves, will get a little bit of priority, maybe a year or two. But after that, other Singaporean researchers will be able to get ready access to the data. There's an expectation that overseas partners will be able to access the data too, subject to them being in partnership with a Singapore entity, so that there's a way of promoting cross-country, cross-institutional collaborations. So it's another cohort on the block. It's another longitudinal population study, but addressing an area of high met need. Multi-ethnic Asian populations have specific research, specific clinical and research questions. Is it going to be fruitful as an exercise? Well, I'm now going to throw you 3 or 4 worked examples, just illustrative examples. Can't do more than that in the time available, just to show that this approach is likely to yield additional discovery that potentially could be valuable. So I'll give you one use case, which is around the carotid atherosclerosis data. From the first, we've quantified the carotid IMT data from the first 10,000 people using standard approaches. We have also in parallel collaborated as an industry collaboration with Metabolon to generate untargeted metabolomic profiling on the first 10,000 people. So we've got dense phenotypic and now dense metabolomic data on the first 10,000 people. We can put up the metabolic variation against the carotid atherosclerosis variation and identify a range of metabolites associated with atherosclerosis, many of which have been previously reported, such as some of the amino acids and the lipoproteins and lipids, but others of which appear to be novel. And we can then start to explore what's the nature of some of these species, and using, for example, Gaussian graphical modeling to pull metabolites into co-correlated networks that allow us to see, well, okay, these sets of markers linked to atherosclerosis probably come, for example, from the carnitine or tryptophan type metabolic pathways. And that includes some previously unknown metabolites labeled X1 and X2 that weren't annotated in the Metabolon database. So identifying co-correlated markers linked to phenotype, but also potentially identifying metabolites not previously annotated. And then we can use our genomic data to build additional confidence by showing, for example, that X1 has got a strong GUAS hit in the carnitine biosynthesis pathway, while X2, previously unknown metabolite, has got a strong hit in the tryptophan metabolite pathway. So we're using convergent metabolomic, genomic data, correlation model building and genetic inference to build out identification of novel metabolites linked to disease that may represent novel biomarkers or parts and causal pathways. Second use case relates to one of my pet interests, the difference in diabetes between ethnic groups. I've shown on the left how South Asians are at highest risk. Within Singapore, there's also tremendous diversity in diabetes risk between the ethnic groups. One of my own research interests has been around using DNA methylation, which effectively integrates environmental behavior exposures with genetic exposures as a singular common pathway. Understanding how DNA methylation predicts future disease, and we've done an epigenome-wide association study. We identify hundreds of methylation markers that predict future diabetes. We can use SMR and co-localization analyses to confirm that the methylation signatures and disease endpoints are likely on the same functional pathway linking a genotype to a phenotype. We can also show that the methylation scores vary by ethnicity, potentially making the methylation scores useful for future predictive value. Okay, moving on. These two next use cases focus on proof of principle that the linkage to electronic healthcare record is going to be valuable. And they're based just on proof of concept, stage 1 proof of concept data, which is a small study, less than 10,000 people. So we wanted to know, can we recapitulate some of the observations you expect based upon published literature? And here puts up polygenic risk scores, reported polygenic risk scores against some important cardiometabolic traits using Ministry of Health electronic healthcare record data. We see, as expected, that polygenic risk scores for LDL, HDL, triglycerides, total cholesterol, and other traits, polygenic risk scores predict the expected quantitative trait in these routinely collected data that you might expect to be murky, but in fact they show nice clean signals. And we can show the same with rare variants too, or uncommon variants. So again, we can show that people with known FH genetic variants have got raised lipid levels in the EHR data. We can also use the same data to start inferring, well, what proportion are on treatment? Turns out to be only, what proportion of people with presumptive pathogenic FH variants are already on treatment? It's only 35%. And so we can start to model out the potential implications for the population from that, which lead to the conclusion that potentially there are 30 to 40,000 Singaporeans with FH in the community, of whom only a third are on treatment, leaving the other 20 to 30,000 at very high future risk for cardiovascular disease. And this then is a really good use case for driving translation to healthcare and implementation of precision medicine vision. Phase 2 is about proving the value of it. It's not trying to deliver it at scale. It's about showing that this precision medicine journey is valuable. So we've got to do that. We have 5 use cases set up as pilot studies. One based upon familial hypercholesterolemia, one based upon breast cancer screening using polygenic and other genomic risk, hereditary cancer syndromes, chronic kidney disease based upon very interesting pharmacogenomic considerations, and then another pharmacogenomic implementation pilot using based around adverse drug reactions. So 5 clinical implementations being launched that have been launched in parallel with each other. Now, one of the design of these studies has been thoughtful. Rather than launching them as 5 individual efforts that might then each individually weakly impact on the ecosystem, the PRECISE has formed a house in which they should collaborate with each other and work together. And importantly, in that house, they need to share resources. They've got a shared kitchen, they've got a shared fridge, they've got a shared living room. And what that means in practice is that rather than trying to generate 5 different genotyping or sequencing facilities, they've got one. Rather than trying to generate 5 or 6 different health economic evaluation platforms, they've got one. And that means that that one genotyping sequencing platform, or that one health economic platform, is stronger, more robust, and is going to be a beacon on which other parts of the ecosystem also start to build their clinical implementation pilots. So it's a way of putting stakes in the ground, strong foundations that, if successful, will add value in subsequent layers of program building. So from my point of view, it's an absolute privilege to have an opportunity within my career to be able to help float and launch this kind of program of work. I think it's a tremendous opportunity that will be great for Singapore, but also great internationally. We've got the strength of bringing Asian diversity. We've got the strength of bringing deep phenotypic and record linkage data. We've got consent that works, and with engagement of the community that brings high recall. We've got fantastic capabilities within Singapore for precision medicine research. And Singapore's a very good, reliable, trusted partner for doing work. You know, when we say, so we'll do something with you, then there's a bond of honor. So thank you for your attention. Last slide. I should thank, this is a tremendous cross-institute, cross-country effort. All the key stakeholders involved. It's not just me, it's not just PRECISE, it's a very large number of people. And that's one of the reasons why it's working so well. Thank you for your attention.